What if a single alteration in your DNA could reshape an entire biological system, influencing how your body manages salt, water, and airflow? Cystic fibrosis offers a clear example of how one disrupted gene can produce lifelong consequences, particularly within the lungs. The disorder arises when changes in the CFTR gene interfere with the movement of chloride ions across cell membranes, creating unusually thick mucus that affects breathing and infection risk. Tracing this condition through history and populations reveals how genetic variation persists across generations, shaped by inheritance patterns and regional ancestries. By examining its molecular origins, global distribution, and evolutionary context, we can understand how a single genetic fault became one of the most widely studied inherited diseases. Cystic fibrosis originates from alterations in the CFTR gene, which directs the production of a protein essential for controlling chloride and bicarbonate movement across epithelial membranes. This transport process regulates surface hydration, influencing the thickness and behavior of mucus in multiple organs. When CFTR activity declines, chloride diffusion slows, water retention decreases, and mucus becomes dense enough to impair airflow and microbial clearance. The resulting environment supports chronic infections and persistent inflammation, forming a central pathway of disease progression. The CFTR protein itself follows a multi-step production sequence, involving transcription, folding, quality control, and membrane placement, variants that interrupt any stage of this process can influence final protein function. Some mutations prevent proper folding, triggering cellular removal before the protein reaches the membrane. Others allow membrane placement but limit channel opening, reducing ionic flow despite the presence of the protein. These mechanistic distinctions explain why individuals with different CFTR variants may experience distinct clinical patterns. Today, classification systems divide major CFTR mutations into functional groups based on their impact on protein behavior. This structure provides a framework for understanding why some variants produce severe respiratory involvement, while others primarily affect pancreatic function. In contrast to many complex disorders, cystic fibrosis demonstrates a strong link between molecular mechanism and physiological outcome, allowing researchers to map symptoms directly to gene activity. Geographically, CFTR variants show concentrated distribution in populations with shared ancestry. Northern European groups exhibit the highest prevalence, reflecting historical founder effects and gene flow across the region. By comparison, populations in East Asia and Sub-Saharan Africa display markedly lower frequencies. These patterns align with known migration routes and demographic expansions observed in ancient DNA records, offering insight into when and where specific variants first appeared. Collectively, these findings illustrate how molecular disruptions, inheritance pathways, and historical population movements shaped the global landscape of cystic fibrosis. Cystic fibrosis genetics centers on an autosomal recessive inheritance model, meaning that individuals must possess two defective CFTR alleles to develop the disorder. Carriers who hold one normal and one altered allele generally maintain adequate chloride transport and remain asymptomatic. This dynamic allows CFTR variants to persist within populations, even though the disease itself affects survival and long-term health. As a result, prevalence is shaped by how often carriers pair within specific communities and lineages. The CFTR gene spans 27 exons and produces a protein composed of regulatory, nucleotide binding, and membrane-spanning domains. Mutations affecting any of these regions can influence protein function. To organize these effects, researchers classify disruptive variants into functional groups. Class I mutations prevent protein synthesis. Class II mutations, including F508del, impair folding and trafficking. Class III variants affect gating behavior at the membrane. Class IV mutations reduce conductance through the channel. Class V and V variants decrease the amount or stability of functional protein available. This taxonomy allows clear mapping between genetic mechanism and Physiological outcome? Today, population studies reveal that CFTR variant frequencies differ across regions. Northern European populations show the highest concentration of F508del, likely reflecting historic founder effects and limited gene flow during early demographic expansions. In contrast, Middle Eastern, South Asian, and African populations display distinct variant profiles, with lower overall prevalence. These patterns illustrate how ancestry shapes the distribution of recessive alleles, and why cystic fibrosis is more common in some regions than others. Ancient DNA research provides further insight by tracing variant ancestry over thousands of years. Early European genomes show evidence of CFTR mutations predating Iron Age societies, suggesting that key variants were already established before major migration waves. These findings show how recessive alleles can persist across shifting populations, and how inheritance patterns follow long-term demographic histories. Cystic fibrosis exhibits clear geographic patterns that reflect long-term population histories. The disorder shows its highest prevalence in Northern Europe, and where carrier frequencies reach notable levels. This concentration aligns with the distribution of the F508del mutation, which became increasingly common during regional population expansions in prehistoric and early historic eras, as genetic drift amplified the allele within relatively isolated communities. It established a stable presence that persists in modern descendants. Today, gradual declines in prevalence occur as one moves southward and eastward. From Northern Europe, Southern European populations show lower carrier frequencies but still maintain recognizable CFTR variant profiles shaped by regional founder events. In contrast, East Asian and Sub-Saharan African populations exhibit markedly lower prevalence, reflecting distinct demographic histories and reduced exposure to the variants that expanded in Europe. These differences illustrate how ancestry influences the distribution of recessive disorders and why specific mutations follow lineage-based patterns rather than universal global trends. In the Middle East and parts of South Asia, localized pockets of higher prevalence appear due to founder variants unique to those regions. These mutations, though distinct from European forms, follow similar inheritance pathways and demonstrate how small communities can maintain characteristic genetic signatures across generations. By comparison, the Americas display a mixture of European, indigenous, and later global contributions, producing varied carrier frequencies linked to regional settlement histories. Ancient DNA studies offer additional clarity by identifying early CFTR variants in Neolithic and Bronze Age samples. These findings indicate that several mutations were already present before major population movements reshaped Eurasia. As groups migrated and mixed, the alleles spread along trade routes, settlement corridors, and expanding agricultural regions. The resulting distribution reflects thousands of years of demographic change rather than a single selective event. Ancient DNA offers critical insight into the origins and early movement of CFTR mutations. Genomic samples from early European farmers and Bronze Age societies reveal that several CFTR variants, including precursors to modern pathogenic forms, were already present thousands of years ago. Their appearance in these early populations indicates that the alleles arose during or before the transition to agriculture, a period marked by increased population density and expanding settlement networks. As agricultural communities grew and dispersed, they carried these variants into new regions, establishing the foundations for modern distribution patterns. Today, lineage analyses allow researchers to trace these early variants across successive cultural horizons. For example, CFTR alleles appear in groups associated with early Near Eastern farming cultures, later moving into Europe during major migration waves. Their frequencies increased within expanding populations, particularly in Northern and Central Europe, where demographic growth and relative isolation amplified carrier rates. In contrast, ancient genomes from East Asia and Sub-Saharan Africa consistently show low frequencies of CFTR mutations, reflecting independent demographic developments and limited contact with populations carrying early variants. As migrations progressed through the Bronze Age, CFTR alleles spread along trade routes and settlement corridors, connecting Europe and Western Asia. Admixture events introduced additional variation, producing regional mutation profiles that persist today. These findings highlight the influence of demographic structure rather than environmental selection in shaping the distribution of CFTR variants. Ancient DNA evidence shows that cystic fibrosis mutations followed the same population movements that defined early human history, offering a clear link between genetic inheritance and long-term patterns of migration. Modern cystic fibrosis research is anchored in detailed analysis of how CFTR mutations alter protein, structure, and cellular behavior. Advances in molecular modeling and high-resolution imaging reveal the precise locations where folding defects occur, how misfolded proteins interact with quality control mechanisms, and why certain variants reach the membrane while others do not. By examining these processes, researchers can identify intervention points that improve protein stability, trafficking, or gating performance. Today, therapeutic development centers on correcting the molecular defects caused by specific CFTR variants. Corrector compounds support protein folding and guide more CFTR molecules to the cell surface. Potentiators address gating defects by improving channel opening once the protein is in place. Combination therapies integrate these mechanisms, increasing chloride movement across epithelial membranes and improving hydration of airway surfaces. These advances mark a shift from symptom-based management to mechanism-targeted intervention. Contemporary research also investigates the relationship between genotype and clinical presentation. Individuals carrying Class II variants often exhibit substantial respiratory involvement, while certain Class IV or V mutations allow partial CFTR function and milder symptoms. Modifier genes contribute additional variability, influencing inflammation, mucus composition, and host defense. These factors help explain differences in clinical progression even among individuals who share the same CFTR variant. Now, new technologies expand the ability to study variant behavior. Organoid models derived from patient cells allow direct observation of therapeutic responses, supporting individualized approaches to treatment. Meanwhile, gene editing technologies explore methods to repair CFTR mutations at the DNA level, offering the possibility of restoring functional protein production in affected tissues. Although these tools remain under development, they provide a clear direction for future research.